Alright, so today we're going to continue with our organic chemistry notes, and we'll be focusing on lipids. So a couple of questions that we want to keep in mind when we're examining these carbon-based molecules is that we want to really pay attention to how they are built, what are some examples of them, and what are their functions inside of the cell. So looking at lipids, first thing that we want to remember is that lipids are made of carbon, hydrogen, and oxygen atoms. Okay? Lipids are also known as fats, and specifically we want to look at the monomer of those lipids. So looking at this molecule right here, you have a glycerol, which acts as the backbone of our monomer of lipids. So you have a backbone glycerol and the fatty acid tail as well. They're actually going to go through some dehydration synthesis similar to how our carbs did to be able to connect with each other. So you can see in your image that you have these OH groups, the hydroxyl groups, that again will do the same process, get rid of that water and be able to link together. Looking at these, you want to recall that the glycerol acts as the backbone and will generally link with their three fatty acid tails to form what's called a triglyceride. Tri for our three fatty acid tails, glyceride for our glycerol backbone. Other names for them, you'll hear them called or named as alls, like cholesterol and estradiol. Okay, those are going to be our different types of lipids. When you're looking at this molecule, you do want to pay attention to the fact that we do have those three carbons here that's going to allow them to link together, and that they can only attach the three fatty acid tails. Okay, because this is mainly made up of the hydrocarbons that we talked about in our last section of notes, this does make it very non-polar. On this end, which means that it's not going to mix with water, H2O. Okay? And so that's a big part of lipids, or big characteristic of lipids. So lipids overall, besides triglyceride, we also have our phospholipids, which we're going to focus on today. And some main jobs of lipids would be energy, for long-term energy use. They're also going to be structural. That's what our phospholipids are going to be used for. And then we just want to keep in mind as one of their big characteristics that they're going to be insoluble in water because of those non-polar fatty acid tails. Okay? For the naming of our lipids, this is really going to be based on the structure of the molecule. And so when we're going to look at these two characteristics. So lipids can be characterized as saturated or unsaturated. And what we're talking about is that they're being saturated or unsaturated with hydrogen. So when a lipid is completely saturated, we're generally talking about that fatty acid tail. If you noticed in the image earlier, we had our carbon chain and completely filled with hydrogen or made with hydrogen atoms. So when a lipid is completely saturated, it's going to be completely filled with those hydrogen atoms, and it's not going to have any carbon-to-carbon double bonds. And the reason why this is important is because it does make the molecule very stiff. It actually ends up being able to lay flat or stack easily. This type of lipid, a saturated lipid, is going to be solid at room temperature. So this a lot of times will be our animal fats, like after you fry bacon and that grease turns to kind of solidify and turns white in the pan. That's going to be our saturated fat. And then for our unsaturated fats, or unsaturated lipids, they are going to contain carbon-to-carbon double bonds, okay? And this can happen once or more than once. So they can be monounsaturated or polyunsaturated lipids. What's important about this is that carbon-to-carbon double bond actually forms kinks. And basically it's bending our fatty acid tail. And when it bends our fatty acid tail, it creates a space in between and actually allows for more movement to occur. So basically this means that this is not lying flat. So it's not going to stack up easily. It's not going to just layer on top of each other. So again, the big important thing about those unsaturated lipids is that they are going to be more fluid, which is really important for our cell membrane, okay? So we have our saturated and unsaturated fats, and those are two ways in which we can name or describe our lipids. We're going to focus in on the cell membrane and the phospholipids themselves, part of that bilayer. So for our phospholipid bilayer, or our phospholipids, you want to pay attention to the two parts of the name. Phospho, we're going to be referring to that phosphate group. And you do need to remember that this phosphate group is charged, or you also remember it as polar. It is polar because it has that charge. And then the lipid portion of it is actually going to be not charged or nonpolar. Okay? Because we have that charged phosphate group and this very nonpolar tail, your phospholipid is actually known as amphihaptic. And this basically describes the fact that it is polar in one area and nonpolar in another region. So amphi is really going to mean that it's both polar and nonpolar. Okay? The reason why this is so important is because this is what allows the cell membrane to form. These phospholipids, because of that charge on the top of the phospholipid head, and you'll also notice that it says hydrophilic head and hydrophobic tail. The hydrophilic water-loving head is going to be on the outside of the cell membrane, so it's going to be basically against that extracellular fluid, or against that cytoplasm, which is made of mainly water. So the head will want to touch the water, whereas that hydrophobic tail, that nonpolar tail, is going to be turning away or wanting to face away from any water, whether that's from the cytoplasm or from the liquid outside the cell or from the cytoplasm inside the cell, okay? And so this is what gives the cell membrane its shape or its structure. Then for functions of our cell membrane, or functions of our phospholipids, they do create that bilayer of phospholipids, so creating our cell membrane. And it is the barrier for every cell. Okay? Another thing you'll want to notice here is that when you have that fatty acid tail being straight, that's going to represent that it's saturated. When you have the kink on your tail, that's going to be unsaturated. So technically this phospholipid could potentially have when it has these unsaturated tails and it has that kink in it, that's what's going to give it this kind of free-flowing motion. It creates space between it that allows it to move more easily because the cell membrane itself isn't static. It's fluid, a fluid membrane, a fluid mosaic model. It's constantly moving and kind of bobbing around there. And the kinks that are being created is going to allow that motion to occur. So then the last little bit here are our steroids, and these are the alls I was talking about earlier. Okay, steroids are just hormones or a type of hormone, and this is going to allow communication by allowing that molecule to actually enter the cell through its membrane. So if you look at this structure right here, this is an example of cholesterol.
Kirkland Smith, come to the front office immediately. Kirkland Smith.
And the cholesterol molecule has that hydroxyl group on it, and the hydroxyl group on it is going to make it polar. And because it's polar, it's going to be able to move into that cell membrane. And we also have right over here this non-polar region. Okay? So the cholesterol, what you really need to know about this is it's going to add to the fluidity of that plasma membrane, okay, of that cell membrane. So, and here estradiol and testosterone are just two more examples of lipids that act as steroids or hormones. You want to make sure that you look over these review questions and answer these, and also take the time to answer all the questions that are on your lecture tools lesson for today.